So we've talked about bacterial reproduction and how that's asexual reproduction, or a basic copy of the original. But then how do bacteria get new genetic information? Because if a bacteria develops resistance to an antibiotic, it should be able to pass on that type of genetic advantage to another bacteria, right? Because otherwise that kind of resistance would just die off. So how does it even do something like that? Well, this is where the plasmid comes into play. So that is the blue little ring of DNA that you're seeing there. So now that we've recapped what we know, let's go into talking about this new method. So we are going to talk about genetic recombination, or ways that bacteria can recombine their genes even though they make direct copies. So let's first talk about a process called transformation. And you can see here that there's two bacteria, and one of them has a plasmid, whereas the other one doesn't have a plasmid. So how could this plasmid transfer over? So transformation is basically where the bacteria takes up naked DNA from the environment. So you can force this type of transfer in a lab by either heating it up or using electricity to cause these breaks in the membranes. And these holes are too small for the larger bacterial chromosome, but it's just big enough for the plasmid to exit, and it floats kind of as naked DNA for a little bit, just hanging out in the environment before it's taken up by another bacteria through those holes that are still there and gets inside. And of course, once you remove that heat or electricity, it then is able to repair its membrane, and then it's a perfectly functional bacteria. So that's transformation. Now let's look at another way that bacteria can genetically recombine. And this is through a process called conjugation. So conjugation is a process where there is a bacteria with a plasmid, and we're going to call this the fertility positive one. So instead of blue, we're just going to redraw this in purple. And F positive means that it's got a fertility factor. So I'm going to write here, fertility factor. And of course, the one without the fertility factor is just labeled F minus. And the bacteria with the fertility factor will create something called a sex pilus. And this sex pilus is just a hollow protein tube to connect the two bacteria. So you can see that this will keep extending and it will connect the two bacteria. And once it connects these two bacteria, and so this plasmid will make a copy of itself, and then it will start moving across the sex pilus, and then it will curl in on itself and form the full plasmid. So you can imagine that this type of plasmid transfer is good for things like antibiotic resistance, because one bacteria can transfer it to another without needing to die or transfer it completely like transformation does. And now there's one last method of genetically recombining, and that is the process of transduction. And transduction is where a bacteriophage, or a virus that infects bacteria, injects its genetic material into the bacteria, and it is either taken up by this plasmid or by the chromosome. So we can pretend that this is scenario 1 and it gets taken up by the plasmid, and this is scenario 2 where it's taken up by the chromosome. So at some point, the virus will lyse the cell and leave after reproducing itself. So when the virus wants to leave the cell, it will need to repackage its DNA. But it not only picks up its own DNA, but it also can pick up either the plasmid's DNA, or on the other case, it will pick up some of that chromosomal DNA. And when it goes on to infect the next cell, it will either bring that plasmid's information or the chromosomal information to be inserted into the next cell. So now you can understand how bacteria can both asexually reproduce and also undergo genetic recombination through these three methods to gain advantages through new genetic information.